So good afternoon and thank you for joining us in today's webinar. I'm Yvonne Lucy. I'm a senior curator at UniProt, and I'm joined by Antonia Locke, my colleague, also senior curator at UniProt, and she will be answering the questions throughout the webinar. So in this webinar, you will learn how to search UniProt database for specific human diseases, proteins of interest, and protein variants. You will learn how to navigate results and protein pages, what type of protein information we provide in UniProt with a specific focus on human disease and variants, and how to use the feature viewer to visualize overlaps of protein variants with protein features and structure. So some of you may already be familiar with UniProt, but I give a short overview what UniProt is. So UniProt is a comprehensive, high-quality and freely accessible resource of protein sequence and functional information for a large variety of species. We provide primary sequences, including isoforms, variants, and post-translational modifications, physiological protein functional information, structural information, including topology and access to 3D structures. And besides the data, we provide also data analysis tools such as BLAST and multiple sequence alignment tools. And importantly, we provide stable identifiers, the UniProt accessions for each specific protein sequence. The scientific literature is a rich source of known information for proteins, and this information is collected through expert manual curation by curators at UniProt. And protein entries are annotated for protein function, including catalytic activities, subcellular location, protein-protein interactions, structural information, including 3D structures, and importantly, also information on protein variants. And for human proteins, we capture information on human disease. For human disease and protein variant information in UniProt, so currently we have 6,523 human diseases annotated in UniProt, which are mostly Mendelian inherited diseases and classified by OMIM. We have over 82,000 missense variants annotated for human proteins, and thereof over 32,000 variants are involved in human disease. We are using the guidelines of the American College of Medical Genetics and Genomics for the annotation of clinical significance of variants to assess the variant category and categorize the variants into benign, likely benign, variant of uncertain significance, and variants that are likely pathogenic or pathogenic. Next to this variant and disease data, we provide tools for the analysis of variant impact on protein structure and function. You can use the feature and variant viewer to analyze the overlap of variants with protein features, including structures from PDB and AlphaFold. And you can use BLAST and align tools to analyze your protein sequence of interest, to find orthologs and identify residues of interest. And importantly, we also provide over 180 crosslinks to relevant databases such as OMIM, DBSNP, ClinVar, PDBE, and AlphaFold. So one of the main activities of our users on UniProt is to search for proteins of interest and protein-related data. In UniProt, you have different datasets available, which are the following: the UniProt KnowledgeBase, or UniProtKB, the species proteomes, UniRef for protein clusters, and UniPark, the sequence archive. The dataset I will focus on today is UniProt KnowledgeBase, which is also the most commonly used to search for protein information, and the default dataset for the free text search bar on the main entry page. There are two types of protein entries in UniProt KnowledgeBase: SwissProt entries and Tremble entries. SwissProt entries are manually annotated and reviewed by one of our expert curators, including sequence analysis and information from literature. The SwissProt entries are tagged with a gold reviewed icon, as indicated on the left. The second section in the knowledge base is Tremble, and this section is automatically annotated based on rules and machine learning approaches that learn from the information in the SwissProt section, but is not reviewed by one of our curators. The Tremble entries are tagged with a gray unreviewed icon. So when would you come to UniProt to look for disease and variant information? And I talk through 3 different scenarios today, how you could search for disease and variant information. First is to search for a known disease and find out which proteins and protein variants are involved in this disease. The second is to search for a protein of interest and find out which disease is the protein involved in. And the third is to search for a specific variant with its variant identifier. For example, its DB-SNP identifier. And I will show you how you can find information about this variant, for example, disease involvement, the physiological function of the protein carrying this variant, and potential characterization of the variant impact on protein function and structure. So let's start first with the first scenario to search for a known disease. So in UniProt, you have different search options available. You can search for a disease or a protein in the free text search bar, where the default search is set to UniProt KnowledgeBase. You can also use the advanced search on the right-hand side, where you can combine search terms with Boolean logic. Another way to find disease information and proteins that are associated with a disease is to search for the disease in the human disease dataset, which you can find under supporting data at the bottom of the entry page. Here, all the human diseases that are annotated in UniProt are listed, including the proteins that are associated with the disease. So by clicking on the Human Disease Access tab, it will take you to the human disease list. Currently, we have 6,523 human diseases annotated in UniProt. And you can find an overview here. The information you can find in the human disease list is the following: the UniProt disease identifier, the disease name, keywords that may be associated with the disease, such as neurodegeneration, as well as cross-references providing links to disease-related databases such as OMIM, MedGen, and MESH. You can then go ahead and find out more about a specific disease, for example, Barth syndrome and cardiomyopathy, by clicking on the disease identifier on the left-hand side. By clicking on the identifier, you will be taken to the disease entry, where you find information such as disease definition, the disease acronym, alternative names of the disease, keywords associated with the disease, and cross-references to disease-related databases. And below the disease description, you will find the related SwissProt entry for the human protein that is associated with the disease described, here human tafacin. At the bottom, you will get a quick overview on the number of isoforms annotated for this protein, as well as protein variants, disease comments, and associated literature added by a curator. Going back to the UniProt entry page, I will show you how to use the advanced search to combine search terms and find information that you are looking for. You can find the advanced search on the right-hand side of the search bar. Here, you can use the drop-down menu to choose your topic of interest that you are looking for. You can, for example, select pathology and biotech in the drop-down menu and choose disease. You can then type in the name of the disease you are interested in, for example, breast cancer. By searching for breast cancer, you will be then presented with a list of proteins that have been annotated having a direct involvement in breast cancer. Here we get 31 proteins involved in breast cancer. To help choosing the protein you are interested in, you can edit the columns in the results table by clicking on the Customize Columns icon on the top of the results page. Here, you can use the search box to look for columns related to human disease and natural protein variants. In the search box, you can start typing the topic you are interested in, and you will be offered with a selection of topics. By searching for disease, you find the column labeled Involvement in Disease under Pathology and Biotech. You can click the box to select it to show you the protein's involvement in disease in the results page as an extra column. You will then find that the column Disease Involvement has been added to your results page here shown on the right-hand side. And you can then go and add the column showing you the natural variants for each protein in the results page by searching for variants in the search box. Now our results table shows us, additionally to the protein and gene name, the organism and protein length, the disease involvement and natural variants of the protein. Of course, you can modify and add additional columns in the results page depending on your topic of interest. So back in our results page, let's pick a protein of interest from our breast cancer search and find out what kind of information we find for this protein. Let's say we are particularly interested in the breast cancer type 1 protein BRCA1 and its involvement in breast cancer. So we can scroll down through the results table to find the entry in the list, or we can search for the entry via the search bar. We can then access the entry by clicking on the protein accession number on the left-hand side of the results table and access the protein page for BRCA1 to gain further insight in its biology. Here on the entry page, we find information on BRCA1, including protein and gene name, status of the entry, here reviewed as SwissProt entry, the organism protein length, protein existence, and an annotation score 5 out of 5, telling us that the protein BRCA1 is well characterized in UniProt. You will then find information on the molecular function of BRCA1 in the function comment, including the references linked with their PMID, the PubMed identifier, to look up the source of information that was annotated by our curators. Further information can be accessed by clicking on the tabs on the left-hand side. In the Topic tab on the left, we can now select the Disease and Variants tab to go to the Disease section for BRCA1. Here in the Disease and Variant section, we find the Involvement in Disease subsection, which provides the information on the role that the protein plays in breast cancer and links to the reference evidences, publications. In this section, a note field is used to describe the role of the gene protein in disease pathogenesis and to distinguish, when possible, between causative, susceptibility, and modifier gene, according to the literature and OMIM reports. In the case of BRCA1, the disease susceptibility is associated with variants affecting the BRCA1 gene. The description field below the note comment describes the disease phenotype, including a link to the online inheritance in men OMIM database with the MIM identifier, where you can find further information about the disease. Below the disease description, you will find a tabular view of any BRCA1 protein variants that have been associated with breast cancer. Here you can find the unique variant identifier given by UniProt, the variant position, the nature of the amino acid change, and the effects of the variation on the protein, the cell, or the complete organism. In the variant description, you will find the disease association, here indicated with the acronym BC for breast cancer, as well as the variant pathogenicity, for example, indicated with uncertain significance. And you will also find, if available, the molecular effects of the variant on protein function and structure, and the literature at the end of each variant line that is describing the variant and its effects on protein function. You will also find a dbSNP identifier for each variant if the variant is captured in the short genetic variation database. And you can click on this dbSNP identifier to go to the SNP database, where you can find further information such as genomic position of the variant of interest, as well as population frequency. You can also click on the variant identifier and you will be provided with further information in an external site on the variant, such as variant position, the variant pathogenicity classification, the residue change, and the physicochemical properties of the residues involved, as well as the variant description. You can navigate the tabs on the left-hand side in this external page to get more information about the sequence surrounding the variant, as well as a list of literature citations describing the variant. So, for example, if you navigate to the sequence information in the left-hand tab, you will be presented with information such as residue conservation between human and various model organism species of the residue in question. And you will also find a presentation of sequence annotations that are captured in the neighborhood of the variant. And these can be post-translational modifications, mutagenesis assays that have been performed in the area around the variant, as well as secondary structure features that have been annotated for this region in UniProt. By clicking on the blue residue change in the variant table back in UniProt, you can then go to ProtVar, which is a tool developed by one of our developers at UniProt for contextualizing missense variants. And here in ProtVar, you can find the following information: the genomic position of the variant, the gene and protein name, and variant information such as disease involvement, as well as CAT and EVE scores. And on the right-hand side, you can see 3 symbols for further annotation for the variant. And you can, for example, click on the primary sequence symbol, and you will be presented with protein functional information that has been captured in UniProt. You can click on the little human for population observations and co-located variants at this position. And by clicking on the little 3D structure symbol, you will be presented with the experimental 3D structure from PDB if there is experimental data available, or predicted structures from AlphaFold. Going back to the BRCA1 protein entry page, I would like to point out that you can find further cross-references to human-specific databases and other relevant resources at the end of the disease and variant section. And here you can find cross-links to human-specific databases such as OpenTargets, OrphaNet, and genetic variation databases such as ClinGen, and chemistry-related databases such as the Drug Compound Resource Campbell. So with that, I would like to come to the second scenario for searching information in UniProt to search for a protein of interest and find out which disease the protein is involved in. For this, I will use again the advanced search and type in the gene name of my protein of interest, here EGR2, and the organism. So I restrict my search to Homo sapiens by adding the taxonomy. By searching in that way, I will then be presented with a list of sequences for the human EGR2 protein that are stored in UniProt. At the top of the list is the reviewed SwissProt entry for human EGR2, labeled with a gold SwissProt entry icon, containing the manually curated information regarding the protein. And the other entries are Tremble entries labeled with the gray Tremble entry icon, and they are unreviewed sequence entries of human EGR2. So we are, of course, interested in the manually reviewed entry that has all the information annotated, and I can access the EGR2 protein entry by clicking on its UniProt accession. So looking at the protein entry for human EGR2, I can read on its molecular function in the function comment at the top of the entry page. And by using the tabs on the left, I can navigate to the disease and variant section to find EGR2 involvement in disease. And by doing so, I find that there are 3 disease comments listed for EGR2. So EGR2 is involved in 3 different types of diseases that are also described by the OMIM database. The first one is a neuropathy, and then the second is Charcot-Marie-Tooth disease, and the third is the Deschênes-Sota syndrome. And for each disease, you will find again the note and the disease description and variants listed under the disease description. I have here shown only the variants for the first disease, neuropathy, due to space limitations. But you will find more variants listed when you go to the EGR2 entry page under the disease and variant section. So going back to the main entry page of EGR2, I have different options to view the variant information that is annotated for EGR2. I can either use the variant viewer or the feature viewer by clicking on the tabs at the top of the entry page, indicated here in the red box. For the variant viewer, you already see that there have been 550 variants annotated for human EGR2. And by clicking on the variant viewer tab, the view will switch from the entry view to the variant view. The variant viewer allows you to look at the protein variants that are annotated for the human EGR2 protein, including the disease variants in a more visual manner. The variants are annotated in a matrix at the top and listed below in a table format. So let's focus first on the variant matrix at the top. The variant view maps all variants on a matrix with the sequence position along the x-axis, indicated in gray, and the amino acid changes on the y-axis. Here you will find the manually curated variants as well as large-scale variants imported from a variety of resources such as ClinVar, Cosmic, Nomad, and Ensemble, and others. On the left-hand side of the matrix, you can apply filters to restrict the view of the variants to a certain type. You can filter based on the consequence of the variant, such as pathogenic variants in red and benign variants in green, or you can filter for the provenance and choose to display only the variants that have been reviewed by a curator, or variants that have been imported from large-scale studies. In the variant table below the matrix, you will find following variant information. The variant identifiers, such as dbSNP, Cosmic, or ClinVar identifiers. The variant position and change, variant descriptions, such as involvement in disease, the clinical significance, and provenance. So I have chosen one of the variants to look at in more detail. You can click on this little black arrow on the left-hand side of each variant to gain further information for a specific variant. Here I have chosen variant at position 4 with an amino acid change from an alanine to a serine. And this variant is associated with Charcot-Marie Tooth disease type 1 and was imported from ClinVar. So by clicking on this little black arrow, you will get more information about the variants, such as the consequence, the variant impact predictions from polyphen and SIFT, the population frequency, the genomic location, the disease association, as well as cross-links to ClinVar, dbSNP, and GNOMAD at the bottom. Going back to the entry page of EGR2, you can then navigate to the feature viewer by clicking on the tab for the feature viewer next to the variant viewer at the top of the page. This will allow you to look at the protein features, including the protein variants, in a more visual manner. So the ProtVista feature viewer allows us to view variants in the context of other protein sequence features that have been annotated for the protein of interest. The ruler on top in gray represents the sequence length of the protein, and the protein features are listed along the protein sequence and grouped in categories. You will find sequence features annotated such as molecule processing, domains and sites, post-translational modifications, mutagenesis information, and the variant information at the bottom just above proteomics and AlphaFold information. And the variant information is here displayed in a distribution profile of variants along the protein sequence. But you can click on the little arrow on the left of the variant tab to see more information about the variants. And just to mention, you can also click on the features, on the feature boxes, and that will bring up the tooltip with information and highlight the sequence position of the feature as shown here for the HCFC1 binding motif spanning residues 162 to 165 of human EGR2. So now we can click on the variant tab to see the variants displayed in a matrix below the features, the protein features. So again, the same matrix is displayed with the variants as we have seen before for the variant viewer. But this time the variants are shown along with the protein features displayed at the top, which allows us to visualize a potential overlap of protein features with the variants displayed. So again, as before, you can filter the variants by the consequence and/or provenance using the filters on the left-hand side. And as we are interested in variants causing disease, let's filter for the pathogenic variants only and filter by selecting the red-colored filter on the left to display the pathogenic variants only. So by looking at the pathogenic variants of EGR2, you can then analyze the overlap with certain protein features. For example, you can select a feature of interest at the top. Here shown the zinc finger domain spanning residues 370 to 392. And if you click on the feature, there will be a yellow bar highlighting the overlap with all the variants that are displayed in the matrix below. So, and you can go and click on the variants to find information about the variants that are lying in the region of this zinc finger that we have selected. Here, for example, I have chosen to show me information for the variant at residue 381, arginine to cysteine variation, and it tells me that this variant is involved in Charcot-Marie Tooth disease type 1D. So with this, I'm coming to the third and last scenario on how to search for disease and variant information in UniProt. And I will go I will show you how to search for with a specific variant identifier to find the variant of interest with a dbSNP identifier and find the associated information for this variant. So I have chosen a variant that is described on the SNP database with dbSNP identifier RS637500001. And I can take this dbSNP identifier and go and search in the advanced search. So in the drop-down menu, you can choose the genetic variation databases and select dbSNP identifier and type in your identifier number. By searching in that way for the dbSNP identifier, you will be presented with one entry in the results page, which is human presenilin 1, which is containing the variant of interest that has been annotated in the presenilin 1 protein entry in UniProt. So by looking for the variant in the variant viewer, you will find all the information that I have described before. So you will find the variant position at residue 435, the amino acid change, and its involvement in Alzheimer's disease type 3. And you also find some characterization of the variant and its impact on protein function. For example, that it almost abolishes gamma secretase activity. So we can then go to the feature viewer to analyze the protein region where the variant has been identified within the presenilin 1 protein. You can go and find your variant of interest in the variant matrix in the feature viewer. And by clicking on the variant, you will be presented again with the information that we have seen before, the residue 435, L2F variation, and the description of the variant involvement in AD3, and the impact on the gamma secretase activity. And by clicking on the variant, you will also see a thin yellow line that is highlighting the overlap of the variant with the features that are displayed above for the human presenilin 1 protein. You can then click on the motif that is in this yellow line, and by clicking on the motif, you will get a pop-up window with further information, and you can find that this motif is the PAL motif, where the variant is positioned. And the PAL motif is described in the associated paper that you can find with the PubMed ID or through EuroPMC. Another paper is describing this region of residue 432 to 434 as important for cleavage of target proteins. And again, the literature is linked to PubMed and EuroPMC. So this analysis shows you a potential impact of your variant of interest on the protein function. In the feature viewer, you can also analyze the effect of your variant of interest on the protein structure. So the feature viewer allows you to visualize the 3D structure of the protein, both experimental structures from PDB, if they are available, or predicted structures by AlphaFold. And you can find them below the variant matrix, the structures displayed, and below is the table with all available 3D structures. You can select one of the structures, for example, the EM structure from PDB. The structure will be shown as well as the variant that has been selected in the variant matrix will be displayed in green in the selected structure. So finally, I would like to draw your attention to the programmatic access of variant data that we offer. You will find the access at the bottom of the main UniProt page under UniProt data, where you can use the FTP download to find the list of human diseases, a file with human missense variants annotated in UniProt, as well as files of amino acid altering variants imported from ensemble variation database. And you can access large-scale variant data via Proteins REST API, including the genomic coordinates of the UniProt sequences. And with that, I would like to acknowledge our funding bodies, the National Institute of Health, Open Targets, Swiss federal government through the State Secretariat for Education, Research and Innovation, and the European Molecular Biology Laboratory Core Funds. And I would like to acknowledge also my work colleagues at the 3 different sites of the UniProt Consortium at EBI, PIR, and SIT in Switzerland. And thank you for your attention.